Thank you for downloading this BJPS short read. BJPS short reads are brought to you by the British Journal for the Philosophy of Science.
To err is not only human. What mistakes mean for philosophy and biology, by David Oderberg, Jonathan Hill, Christopher Austin, Ingo Bojak, François Cinotti, and John Gibbins. Read by David Oderberg. Concepts such as purpose and goal-directedness have an uneasy history in the philosophy of biology. They're teleological concepts, and teleology harks back to a pre-modern, quasi-Aristotelian understanding of biology. Biologists themselves use teleological language liberally, yet philosophers tend to be warier, perennially looking for ways to naturalize teleology. And this means roughly explaining it without invoking the so-called spooky metaphysics of vitalism, or the final causes that are supposed to exert their power from the future to the present, or the invisible hand of an intelligent mind directing organisms' behavior. It is, however, possible to analyze the goal-directed, purposeful behavior of living things in ways that are both amenable to empirical investigation and philosophically plausible without scaring the horses, as it were. Indeed, the global multidisciplinary Templeton Foundation research program, Agency, Directionality and Function, of which our team is a part, has this aim in mind. Our own line of research concerns the concept of mistake-making. We think this phenomenon is a universal feature of organisms, species, and even many parts and subsystems of organisms. It's not only humans who make mistakes. The cat that jumps on a shrew thinking it's a mouse and then spits it out. The domestic hen that tries to hatch a golf ball. The fish that takes the bait. The frog whose darting tongue misses the insect by millimeters. These are all examples of organisms getting things wrong. In other words, making a mistake. It is clear that what they were trying to do is not what they ended up doing, in a minimal sense of trying that does not necessarily require any effort or self-reflection. And what they ended up doing is not the sort of thing they're supposed to be doing as far as their own intrinsic natures are concerned. That, broadly, is what we mean by making a mistake. Think of the liberal use of mistake-related terminology even at the microscale and at the subsystemic level. Bacteria can be confused by plants mimicking signals used by the bacteria for infection of the plant. Antibodies can be fooled by pathogens. Competitive inhibitors can trick an enzyme into binding to the incorrect substrate. Just how close to universal mistake-making is in biology is an exciting project for biologists to consider. We hypothesize it might even be found in blood platelets if they can be fooled by collagen-like proteins, instead of collagen protein itself, into beginning a blood clotting process that is not supposed to happen and could even be life-threatening. Philosophically, it's vital to distinguish mistakes in general from the mistakes typically made by humans. Such mistakes are often conscious or can be brought to consciousness through reflection, and involve moral concepts such as carelessness and recklessness, as well as issues to do with free will, responsibility, accountability, and so on. We claim it's possible to work our way outwards from what is typically human to an extended concept of biological mistakes in general, of which human mistake-making is a distinctive instance. Moreover, what we find of particular importance in the concept of biological mistakes is a way of operationalizing teleology, namely, making it the subject of empirical investigation, turning it into material for the generation of novel, testable hypotheses of interest to the working biologist. We set out some of these ideas in a recent paper and developed them in our current BJPS article, providing several examples. For instance, researchers have described working on cell decision-making errors resulting from noise and signaling failures. They found that a cell can respond differently to the same input, leading to incorrect cell decisions and responses. One kind of mistake is a false alarm event, where there is no object, such as a particular protein, but noise leads the system erroneously to declare the presence of that object. Another is a misevent, where the system misses the presence of the object. The likelihood of a false alarm or misevent can be computed using probability distributions, but our mistakes framework leads us to ask the following questions. First, is there a cellular mechanism for distinguishing signal and noise, thus enabling direct investigation rather than indirect via probability distributions? And second, can cells mismeasure the amount of a given protein in this kind of decision-making situation? And if so, what is the underlying mechanism? We do not suppose that for a given hypothesis involving mistake-making, a biologist could or would not have come up with that hypothesis without appealing explicitly to the theory of biological mistakes. Rather, our theory is a way to organize many of the thoughts and concepts experimentalists naturally work with when they generate hypotheses for testing. But also, we hope, the theory of biological mistakes will encourage biologists to consider phenomena that may not have previously occurred to them. What mistakes does their target organism typically make? What potential might it have to make other kinds of mistake? What underlying mechanism might there be by which a certain kind of mistake is made? And how common is it, if at all, across a variety of organisms of widely different kinds and at different scales? How does their organism avoid mistakes or correct them? And if it cannot, why not? How does mistake-making vary with the kinds of trade-off an organism may make between, say, timely action in its environment and sufficient or effective action, say, to avoid a predator or find a mate or repair damage? It is exciting that the mistakes framework is capable of generating many ways, including, we believe, novel ways, of understanding and testing a system. That said, difficult conceptual questions need to be resolved in order to firm up our framework. For example, there is a broad sense of mistake that encompasses what we investigate in our present BJPS article. Included in this would be all states, conditions, structures, processes, and so on that deviate from what we call a standard of correctness for a system, say by undermining an organism's health or bodily integrity or survival. However, we focus on the making of mistakes. Strictly, every mistake is made, and this requires a concept of agency by which the behavior of a system, organism, or part, or cell, species, or group can be said to amount to the making of a mistake. There is, we claim, a metaphysical difference between mistake and mere failure. It is a mere failure to be hit on the head by an unexpectedly thrown rock. It is a stupid mistake to try and eat a rock. Mistakes are things we do. Failures are what simply happen to us. We develop a rigorous concept of what we call the minimal biological agent to accommodate this need, a concept that applies as much to the actions of enzymes as to those of insects or birds. We also need, of course, a rigorous definition of biological mistake, and this we propose as well. Our basic idea here is that a mistake is something done by an organism or similar that threatens its effective action in its environment. We adopt a liberal concept of threat, according to which an act is a threat if, when not mitigated, it undermines further effective action, or function in a broad sense. A deer that walks in front of an oncoming car is subject to a threat. If the car doesn't stop or the deer doesn't move fast enough, it will be injured or killed. A person who ingests ethylene glycol has made a potentially fatal mistake, but so has the alcohol dehydrogenase enzyme that binds to it, since the enzyme's binding action is a threat to the organism to which it belongs. This threat can be mitigated by introducing ethanol into the system, which is a competitive inhibitor to ethylene glycol. It reduces or removes the toxicity, but when the enzyme binds to the ethanol in this situation, there is no threat to the system. Presumably, the medical worker mitigates it by controlling how much ethanol itself is introduced. It would, however, be a mistake for the enzyme to bind ad libitum to ethanol, as the effect would also be toxic. Yet an enzyme has no awareness of what it does. It just acts. And the relevant standard of correctness is that enzymes act for the benefit of the organism to which they belong, according to that organism's specific needs. When they fail to do so, they err, yet without necessarily malfunctioning. Can enzymes be trained or conditioned to bind only to certain substrates for the system's benefit, and not others that will lead to harm, even if the geometry is perfect for both? If binding to the wrong substrate is a case of being fooled, how do enzymes protect themselves from binding to incorrect substrates willy-nilly, given that exposure to them is likely to be common in many typical situations? We think mistake theory can provide a structure within which these sorts of questions can be systematically investigated.